Hello, and thank you for being here. My name is Edoardo Sarti, and I will present my work titled Paralog Function Annotation with ProfileView. This work has been done in collaboration with Alessandra Carbone, Julien Henry, and Théo Lemoigne, all at the LCQB laboratory at Sorbonne University. The team led by Julien Henry is doing an extensive effort for determining the structure of all the 11 proteins participating in the Calvin-Benson cycle in Chlamydomonas reinhardti, a unicellular algal model organism. The Calvin-Benson cycle is one of the main chemical pathways present in all photosynthetic organisms. Its role is to fixate the carbon present in CO2 molecules into glyceride 3-phosphates or G3P molecules, which ultimately will be used to produce glucose. Due to the long evolutionary history of this pathway, each protein has several paralogs that share many common sequence features but play different functional roles inside the cell and do not contribute to the Calvin cycle. The problem is thus, for each species, how do we find the correct paralog that is involved in the Calvin-Benson cycle? This task requires knowing the function of each protein, but functional annotation of paralogs is notoriously difficult both with specific experimental assays and with large bioinformatics studies. Recently, a new method for function prediction called ProfileView has been published by the team of Alessandra Carbone at LCQB. The algorithm is based on the use of hidden Markov models and hierarchical clustering, and does not need any multiple sequence alignment. For building the library, ProfileView considers a set of seed sequences provided by the user, and for each one of them it searches into a sequence database for hits with very high sequence identity. A hidden Markov model is thus built for each query sequence and added to a library whose scope is to store profile information about the seed sequences. When the library is built, the user enters the protein sequences that need to be functionally classified. At this point, a classical phylogenetic algorithm would consider their distance in the sequence space and build a phylogenetic tree with a hierarchical clustering method. Instead, ProfileView embeds the sequences in a representation space defined by the HMMs contained in the library. This space has one dimension for each HMM. When a sequence is aligned against an HMM, a likelihood is returned, estimating the probability of that sequence to have been produced by the HMM, that is to be similar to the group of sequences the HMM was trained on. Thus, each query sequence is aligned against all HMMs, and the resulting vector of likelihoods is interpreted as the coordinates of the query sequence in representation space. Finally, hierarchical clustering is applied in order to obtain a dendrogram, which will be based on this functional space. ProfileView has reached excellent performances in predicting the function of homologous proteins, but it has never been used for paralog function annotation before. We thus have used a new protocol in order to maximize the signal. For each Calvin cycle protein, a list of pertinent PFAM domains is extracted. We start from the PFAM full multiple sequence alignment and on subsequent steps of clustering with MM6 for choosing around 1,500 sequences with which to build the model library. These are thus sequences relative to functionally important domains. Indeed, considering full protein sequences might introduce unwanted noise. Also, there is no taxonomic selection on the sequences used for building the library. A remarkable fraction of them comes from eukaryotes and other non-photosynthetic organisms. This is found to facilitate ProfileView in its functional classification, as it provides a more consistent set of negative examples. Then, the maximal set of photosynthetic homologous sequences is entered in ProfileView in order to be classified. For each protein, we have manually curated the annotation of a set of sequences in order to see whether Calvin-Benson and non-Calvin-Benson paralogs are correctly classified in separate subtrees. This is the result of ProfileView analysis on the fructose 1,6-biphosphate aldolase, FBA, where we can clearly see the Kelvin-Benson paralogs being clustered in a different subtree with respect to the non-Kelvin-Benson ones. We obtained comparable results for 8 out of 11 proteins. Out of the remaining 3, the method seems to not discriminate in the case of Rubisco, but this is actually expected because Rubisco doesn't have any paralog. In the other 2 cases, ProfileView does differentiate Kelvin-Benson and non-Kelvin-Benson proteins, but the data points happen to be in a very limited region of the tree, and further analyses are needed. In summary, Kelvin-Benson proteins have multiple paralogs, and it's hard to guess which one will take part in the cycle. ProfileView correctly classifies the function in 8 out of 11 Kelvin-Benson cycle protein families, and the preliminary results show that the functional classification we produced can be used for protein-protein interaction prediction. Thank you for your attention.